Hey guys, welcome to the third part of your Unit 1 notes. We're going to continue to talk about research methods that are used in psychology. Remember that if you do end up majoring in psychology in college, that you most likely will have to take at least 2 courses that go back to research methods. Our first part is experimental research. So we want to find out if there's any sort of cause. Is there a relationship between what you're looking at? It's really important that it's truly randomly assigned. And again, you need to have your independent and dependent variables. Independent is manipulated. Dependent does not change. So D and D. Dependent does not change. It's what's being measured. In your experimental group, you have the independent variable, which does change, get altered, and then your control group is treated equally, but there's no manipulation of the independent variable there. So it's what you kind of compare it to. We also have to make sure that we have validity. Is it actually reflective of what's happening in the real world? Do the results that we get from certain experiments apply to real-world applications? So that's our external validity. Internal, are the dependent variables changing the results of the independent? Are there any biases? Is there a logical error? So when we do have these experiments, we have to do them multiple times to ensure that there is validity. Next, we have a few of our biases and expectations. Experimenter or research bias is the scientist or whoever's performing the experiment kind of is like, I really want this to happen, and kind of sways the results to do so. Demand characteristics, participants form an interpretation of what the experimenter wants and kind of changes their behavior to fit that way. Research or subject participant bias is the subject consciously or subconsciously acts the way that they think the experimenter wants them to act. So kind of like demand characteristics, but sometimes they don't even know they're doing it. Placebo effect is a treatment or substance that has no effects. So if I wanted to test a pill that makes your right eye blink, I would give some people the pill that makes your right eye blink and some people a placebo pill. So it's a sugar pill, but they think it's going to make their right eye blink. Double-blind experiment. So neither the participants or the experimenters know who's receiving a treatment. So if I was the experimenter and you guys were my test subjects, I would have a third-party person administer whether or not you get the placebo or the pill to make your right eye blink. Single-blind experimenter is the researchers and those involved in the study do not tell the participants if they're getting a certain treatment or not. If any of you guys have watched Grey's Anatomy, they've done quite a few experiments in the later episodes where they did a double-blind experiment, but then we all know Meredith peeked at one and that caused it to become a single-blind experiment. Research sample. Population is looking at the entire group of who we're going to draw from. So if we say all of Northern High School. Sample, a portion of that population who is actually observed. So if I say I want one person from each classroom. A representative sample are characteristics to that population. So if I want to compare all the schools, I might ask each school to allow one person to be in my study from each classroom. And then a random sample, there's just an equal chance that you could be selected. So if I know I'm going to each classroom, I would put all of one teacher's students in a hat and then mix it up and draw it, and that's the one person in my sample. So there are two sort of settings within our research methods, we have the laboratory setting, which is controlled. You can control almost anything. Temperature, amount of light, who goes into the laboratory, who doesn't. And then we have our real world. So if you remember back into our last video, we talked about naturalistic observation. We're in our natural setting. We can't control everything. So I want you guys to answer this question. What do you think are some of the advantages and disadvantages of each of these settings? And I want you guys to put a star by the advantages and disadvantages of each setting. Make sure you guys do write this in a complete sentence. So once we do have our information, we're going to then need to analyze it and interpret the data. This is where statistics comes in, and if you guys are an AP Stat, great. This will be very easy for you. If not, we're going to the very basics of stat. But it's the part of math we need to use to report the data. We have descriptive statistics, which describes and summarizes the data. You guys should know your mean, median, and mode from like 3rd grade. Mean is your average, median is the middle number, mode is what happens most often. Range, you guys know it's the gap between the lowest and the highest scores, and standard deviation is how much the scores deviate from one another. Descriptive versus inferential statistics. Inferential draws conclusions. Do we have confirmation that the hypothesis we predicted is correct? Is there statistical significance? And do we see a relationship between the sample and the population as a whole? And we'll get more into that. Then we have our normal bell curve, and this is where you guys see the standard deviation. The normal bell curve is typically a symmetrical bell-shaped distribution of scores. Could be grades, weights, heights. But this bell is occurs so often that we have now labeled it the normal curve because we see it happening so often. So on your paper, I want you guys to this question there and answer it. What is the difference between descriptive statistics and inferential statistics in your own words? Okay? In your own words, what is the difference? So based with psychology, descriptive statistics uses the data to provide descriptions of the population. Graphs, calculations, tables. Inferential makes inferences and makes predictions about population based on the data from those populations. Then we have our research ethics. So nowadays we have tons of research going on, but we have to make sure that the participants have their rights. So the APA came up with the Institutional Review Board, and they have to go through and approve or disprove everyone's experiment and their designs. APA guidelines do say that you have to have consent. There has to be confidentiality. You have to debrief after an experiment is run and then talk about deception. So back in the earlier years of psychology, they just said, hey, I'm doing this experiment, great, let's move on. Nowadays we can't. I'm pretty sure if you guys go to be in an experiment, you will see that you have so much information in the beginning. It's like saying, hey, this is what we're planning on looking for. You could be in this group or this group, and here are all of your rights. But then we also know that it's not just humans that are part of research ethics. We also have animal research. And we know that animal research has benefited humans drastically. However, it's only used by 5% of the researchers. And typically it's usually rats and mice used 90% of the time. But because of the IVR, we have to have very high standards for these animals. So their housing, where they're kept, needs to be clean. They need to be fed appropriately. And they can't be misused or mistreated. So they make sure their psychological and physical well-being is being kept. So if you guys got through all these, make sure you draw a little tiny rat to the best of your ability on the bottom of these notes. And I will see you tomorrow. Have a great day. Bye.